we talk about the control of respiratory function. The control of respiratory function is designed to do really two main things, okay? And when we talk about the control of respiratory function, we're really talking about the involuntary control of respiration, the sort of level of control that is beyond our conscious awareness, okay? Now, when we talk about respiratory activity is quite rhythmic, right? There's cycles of inspiration followed by expiration. And we talked about how the muscles that control the inhalation include the diaphragm and the intercostal muscles, the external intercostal muscles, and then the forceful breathing, we get the neck muscles involved, we get the internal intercostal muscles involved, and the abdominal muscles, right? Now, the intercostal muscles are controlled by the intercostal nerves. Diaphragm is controlled by the phrenic nerve. Now, the cell bodies of the cells within the phrenic nerve, okay, the neurons within the phrenic nerve, the cell bodies are actually sitting in the spinal cord very high up in the cervical spine. It's sitting at C3, 4, and 5. Okay? And it emerges out of the spinal cord at cervical level C3, 4, and 5, while the intercostal nerves come out of the spine throughout the thoracic spinal cord. Okay? Now, the fact that the phrenic nerve innervates the diaphragm, and the diaphragm is critically important for breathing. Okay? So when you hear about people with spinal cord injuries where they actually lose the ability for spontaneous respiration and they rely on mechanical ventilation, this is when their spinal cord injury is very high up. This is when their spinal cord injury is above levels C3, 4, and 5. Okay? Because a spinal cord injury below C3, 4, and 5 means that the phrenic nerve has already emerged out of the spinal cord and is unaffected by the spinal cord injury. Okay? So individuals that have spinal cord injuries that are high up, above C3, 4, and 5, are going to lose the voluntary contraction of their diaphragm and involuntary contraction of their diaphragm, and are going to be reliant on mechanical ventilation. Okay. Now, if you go farther up and go to the brainstem and look at the medulla and the pons, what you find is basically the respiratory center of the brain, the area of the brain where all respiratory activity, involuntary control of respiration, originates. Okay? So among these different areas, we have a couple of areas in the medulla, and together we call them the medullary respiratory center. And then we have two areas in the pons that we sometimes call the pons or the pontine respiratory centers. Okay. Now, the most important area for control of respiration is really the medulla, right? Is really within the medulla. Okay. So within the medulla, we have these what are called nuclei, which are areas of where there are a lot of cell bodies. Okay? And these nuclei, there's a nuclei that lies dorsally, called the dorsal respiratory group. There's a nuclei that lies ventrally, called the ventral respiratory group. And then we've got one on top called the pre-Boltzinger complex. Okay? Now, the dorsal respiratory group directly controls the muscles involved in restful breathing. Okay? So the dorsal respiratory group controls the muscles involved in restful breathing. And what are those muscles? Diaphragm, external intercostal muscles, and that's it. Right? That's restful breathing. Okay? You contract them when you want to inhale, you relax them when you want to exhale. Okay? When you start to forcefully breathe, you start to recruit your ventral respiratory group, because the ventral respiratory group controls the accessory muscles for both inhalation and exhalation. Okay. And then what the pre-Boltzinger complex does is maintain or control the rhythmicity of breathing. It controls the respiratory rate. Okay. So sort of the direction of control, it starts at the pre-Boltzinger complex. The pre-Boltzinger complex for restful breathing is triggering the DRG, the dorsal group, and then the dorsal group is rhythmically stimulating those muscles. Okay, so you start pre-Boltzinger complex, DRG, and then the DRG stimulates the muscles. Okay, pre-Boltzinger complex, DRG, muscles. Now, when you want to forcefully breathe, now the direction goes from pre-Boltzinger complex, DRG, to VRG, to the muscles. Okay? So the DRG recruits the VRG. So you start using your accessory muscles. Okay? So for right now, for all of you that are breathing restfully, if you looked at your medulla, you'd see rhythmic activity in your pre-Boltzinger complex, rhythmic activity in your dorsal respiratory group, and pretty much no activity in your ventral respiratory group. Okay, because you're not effortfully breathing. Okay? If I asked you to start to run around the room, everything would start to light up, right? Because you'd start to forcefully breathe. Okay? Now, so what does the pons do? What do these two centers in the pons do? What they do is they start to adjust. They regulate breathing. They don't directly control it, they just regulate it. And they will adjust the rate and depth. Okay? Bless you. They'll adjust the rate and the depth. Okay? And the way they really do that is by controlling the duration of your in-breaths. Okay? So if I am increasing the duration of my in-breath, right, that means I'm spending more time during the inspiratory cycle than I am during the expiratory cycle. And what's that going to do to the depth of my breathing? It's going to increase it. And what's it going to do to the rate of my breathing? Slow it down. Okay? So by controlling how long I'm inhaling, the pontine centers are adjusting the rate and depth of my breathing. And in general, the pneumotaxic center, the one on the top, tends to prolong the in-breath, while, excuse me. The pneumotaxic center tends to shorten the in-breath, while the apnoistic center tends to prolong the in-breath. Okay? So pneumotaxic center tends to shorten the in-breath. Apnoistic center tends to lengthen the duration of the in-breath. So both are usually active at the same time, sort of fine-tuning the rate and depth of breathing. Okay? Now, if you are just not thinking about your breathing, and the rate and depth of your breathing is changing, why is it changing? What needs or what changes in your body are going to cause these changes? So I heard somebody say the oxygen needs, right? And the oxygen needs are really about the metabolic needs of your body. Okay? It's really about the metabolic needs of your body. Okay? So why is it that I start to breathe more rapidly and more deeply when I'm climbing stairs? Because I'm contracting muscle, my rate of oxygen consumption is increasing, my metabolic demands are increasing. So I need to get more oxygen in my body and deliver it to tissue. And I do that by increasing rate and depth of breathing. Okay? So the pontine center and the medulla is receiving sensory input from the body about metabolic demands. Okay? And it's adjusting breathing based on those metabolic demands. Okay? When you're sitting around, relaxing, watching TV, your metabolic demands are very low, so your breathing is going to be it's going to be at a low tidal volume, low depth, and low rate. Okay? As soon as you start walking around your apartment, you start, doing something, your metabolic demands get a little higher, you start to increase your rate and depth of breathing. Okay? And as you start to exercise, demands go up even more, and rate and depth go up even more. Okay? So there is a top-down control of breathing. Okay? There is a top-down control of breathing, and then that top-down control is adjusted based on sensory information coming back from the body about metabolic demands. Okay? And that's all sort of involuntary control. You don't have to think about breathing harder when you're exercising. Okay? In fact, you can't stop breathing harder when you exercise. Okay? Because it doesn't matter what you want. The brain gets what it needs, okay? And the body gets what it needs. Okay? Questions? Yes, ma'am.
How does the regulation by the pontine center differ from exactly what the pre-oxygen?
The difference between what the pontine centers do and the pre-Boltzinger complex does is that the pre-Boltzinger complex, it's just a given frequency, right? It's just a static frequency, and it's a natural firing rate for those cells. The pontine centers are going to change that. It's going to modify that based on what it sees that the body needs. Okay? Yes, ma'am. Yeah, so the pre-Boltzinger complex is a group of cells that fire action potentials at a natural intrinsic firing rate. So do you remember the SCN, the suprachiasmatic nucleus? It had a natural firing rate that varied in 12-hour increments. The pre-Boltzinger complex fires whatever it is, 10, 12 action potentials per minute all the time. And it just, every time it fires action potentials, it stimulates the DRG. The pre-Boltzinger complex only goes to the DRG, And then the DRG is what goes to the VRG if necessary. Sensory information coming from the body. Yeah. So when you start to exercise more or use more energy, what happens is things like your CO2 level goes up. That information goes to your brain and literally to your pons and to your medulla, and it tells the DRG we need to stimulate the VRG. Yeah. Yeah.
I was just going to ask about that. So it's actually the need to go off the CO2 and bring acidity down as the driving force.
So that's actually the topic of most of the rest of the lecture. So what type of sensory information is coming in is the type of sensory information is the PCO2 of the blood, the O2 of the blood, and the pH, right? So all three of those are going to come back into the brain and affect breathing. And I'll talk about sort of the relative importance of those three, but yeah. Okay. When we look at respiratory control, the essence of respiratory control is, number one, that respiration is under both involuntary control and voluntary control. Okay? And that involuntary control of respiration is an interplay between basically top-down control, okay, and feedback from the body regarding metabolic needs, and that adjusting the rate and depth of breathing. Okay, so what's a normal respiratory rate for a resting state? 10 to 12, 10 to 15, somewhere around there, right? And usually the tidal volume or the depth of breathing in a restful state is pretty minimal. It's about 500 milliliters, right? Half a liter. So breathing restfully, breathing normally, you're breathing about 10 to 12 breaths per minute at around 500 milliliters per breath, okay? Now, if you started climbing a flight of stairs, okay, your respiratory rate would go up and your respiratory depth would go up. So tidal volume would go up and respiratory rate. And that's because your metabolic demands have changed, right? When you're climbing up a flight of stairs, you need more oxygen to fuel muscle contraction of the legs, right, so that you can climb those stairs. So as your metabolic demands change, your respiratory rate and depth also change. Okay? So we have control of breathing in terms of the efferent control, and that is originating at the medullary respiratory group, being fine-tuned by the pons, okay? And then the feedback coming into the brain in terms of metabolic demands is adjusting the rate and depth of breathing, okay? Now the metabolic feedback that's coming in, and we're going to talk about this, comes basically in three forms. There's the PO2 of blood, the PCO2 of blood, and the pH of blood. Those are the three pieces of information that your brain is constantly getting that is helping to adjust breathing. Okay? So you notice that the respiratory center is entirely in the brainstem, which means if you have loss of brain function above the level of the brainstem, okay, everything about you would cease to be, right, in terms of your personality, in terms of your voluntary control of anything, your language, right, everything would cease to be, but you would continue to breathe, okay? You would continue to breathe, and as long as your heart continued to get oxygen, your heart would continue to pump. Okay? Now, so that means that if you have removal of all of the brain, with the exception of the brain stem, breathing would remain normal. Okay? Now, if you removed input from the pons, okay, if you removed all input from the pons, so you made a cut between the pons and the medulla, but you maintained input from the body, right, sensory input from the body. Again, the respiratory rate would be maintained. Respiration would be maintained. And it would be constantly adjusted for the rate and depth of breathing, right, in terms of metabolic demands. Okay? If you, no matter what you did to the brainstem, if you removed the feedback to the brain, like you snipped both vagus nerves, so you have no sensory feedback to the brain, okay, in terms of metabolic needs. What would happen is you would have a regular respiratory rate, but if your metabolic demands changed, your respiratory rate would not respond. It wouldn't change. Okay? So if the brain receives no feedback about metabolic needs, it just maintains sort of a baseline respiratory pattern. Okay? Now, there is a breathing pattern that is described as apnoesis. And apnoesis is a respiratory pattern that it tends to occur when you have severe brain trauma. Okay. And what it looks like is basically these long gasps followed by short exhalations. Okay. So breathing becomes a series of these long gasps. Okay. So again, this is usually a result of pretty profound brain injury. So the thought of this is that the brain injury is doing two things that's causing this apnoesis. It is diminishing the input from this pneumotaxic center. Okay. It diminishes the contribution of the pneumotaxic center, which ends up increasing the dominance of the apnoistic center. Okay. If you recall, those two centers tended to affect the duration of the in-breath in opposite directions. Right? Apnoistic center tends to increase it, pneumotaxic center tends to decrease it. Okay? So basically, something has happened that's caused the apnoistic center to become dominant. But then on top of that, what also must be happening is that there is an interference in the brain's ability to either receive or interpret the sensory input coming in from the body. Okay. Because the sensory input should be enough to maintain a normal respiratory rate. But when the brain either can't receive that signal or it can't interpret the signal because of the brain injury, you get all top-down control. Okay? And you end up with these gasps because the apnoistic center is prolonging that in-breath. Okay? So we're going to talk about this sensory feedback that comes back to the brain on metabolic demands and how what conveys these signals and how it affects breathing. Okay, so we're going to talk about that. But before we get into that, I want to talk about one pretty small topic in terms of respiratory control. And that is the control of respiration that is designed specifically to protect the lungs. Okay, so these are a variety of pulmonary receptors. Okay, so sensory afferent nerves that are located within the lungs and are adjusting the rate and depth of breathing in order to protect the lungs. Okay? These guys don't really care about metabolic demands. They don't really care about normal breathing patterns. Okay? They're trying to protect the lungs from injury. So these pulmonary receptors are responsive to basically two types of stimuli, stretch and irritation. Okay? So stretch happens when the lungs inflate, right? When the lungs expand, the airways open up, right? They dilate, and you get stretch. Okay? Now, stretch is important, obviously, in order to ventilate the lungs. But if stretch happens too rapidly or to an extent that's too much, you can get overinflation of the lungs and injury of lung tissue. Okay? So the stretch receptors are trying to prevent overinflation injury. And the irritant receptors, they sense the presence of inhaled irritating substances, like ammonia, like smoke, right? And the irritant receptors are trying to protect the lungs from chemical injury. So what's interesting about these receptors are that I'm going to talk actually about 3 different sets of pulmonary receptors. And all 3 of these sets are sensitive or can respond to both stretch and irritation. Okay, so all of the pulmonary receptors I'm about to talk about are capable of responding or being sensitive to both stretch and irritation. But some of them are more sensitive to stretch, and some are more sensitive to irritation. Okay, so they got a day job and they kind of have a side job. All right. So we have a group of receptors that we call the pulmonary stretch receptors. They're actually found in the bronchiolar smooth muscle. Okay, they are highly sensitive to stretch, and they're sort of minimally sensitive to irritation. And these pulmonary stretch receptors are responsible for a reflex that often happens at the onset of exercise. It's called the Herring-Brewer reflex. And basically, you especially if you're not warmed up, if you just start, you go from 0 to running, right? Especially if you're me, what you end up doing is you're doing a lot of gasping, right? So what the Herring-Brewer reflex does is when I try to immediately start exercising at a pretty high level from 0 to high, you start to take in a couple of really big breaths, right? You start to get kind of air hungry, you take in a couple of big breaths, and it triggers these pulmonary stretch receptors. And what happens is they trigger this reflex, this Herring-Brewer reflex, and it causes breathing to become more shallow and rapid. Okay? And again, it's designed to protect the lungs from overinflation. Okay? So onset of exercise, you take in a couple of really big breaths, this reflex kicks in, and then all of a sudden you start taking in more shallow but rapid breaths. And then eventually your breathing might normalize as you warm up and get into the exercise. All right. The pulmonary irritant receptors, as you might imagine, are very, very good at highly sensitive to the presence of irritation or inhaled irritants, and less sensitive to stretch. Where these guys live is actually in the lumen of the airway. Okay? So these receptors are found dispersed among the epithelial cells that line the airway. And the epithelial cells that line our respiratory tract are a particular type of cell called the columnar, ciliated columnar epithelial cell. It's called columnar because the cells look like they are big columns, right? And they're ciliated, meaning that they have these little hair-like projections called cilia. And those hair-like projections are constantly waving about in order to help sweep mucus out of the airway. Okay? So dispersed among those epithelial cells are these pulmonary irritant receptors. Okay. Now, when they sense the presence of an inhaled irritant, like ammonia, like smoke, okay, what they do is they cause a reflexive bronchoconstriction, okay, and a breathing pattern that looks a little bit like a gasp. And the reflex is designed to minimize the exposure to the inhaled irritant. Okay. So you get constriction of the airways and kind of this gasping breathing, okay, to minimize the exposure. The third type of receptors, I don't have a separate slide for them, but they are also irritant receptors. They're called J receptors. J as in Jack, okay, J receptors. And actually that J stands for juxtacapillary. Okay, juxta in the physiology world means juxtaposed or right next to. Okay. Because those J receptors are actually found within the alveolar sacs, very close to the sites of gas exchange. Okay. And again, they are irritant receptors, and they trigger much the same reflex as the other irritant receptors, bronchoconstriction and gasping. But those J receptors are really concerned with the any damage that might happen to the sites of gas exchange, okay? Any potential absorption of that irritant into the blood. Okay. Yeah. So the J receptors, because they're located in the alveolar sacs, they do the same thing that the other irritant receptors do, but they are primarily concerned with preventing damage to the site of gas exchange and preventing the absorption of potentially a harmful substance into the bloodstream. So if you ever, as a youngster, tried to look cool and smoke, what does somebody look like when they smoke for the first time? Yeah, their chest tightens up and they start to cough and they can't catch their breath, right? And that's exactly what you're looking at when you're with they're activating these irritant receptors like crazy. Okay, so you just got to push past that to get to the good part of smoking. Yeah. Yes. Yeah. And hopefully you are. So the question was, is that triggering both the irritant and the J receptors? Yes, but hopefully you're triggering the irritant receptors first, and it's preventing the substance from getting to the site of gas exchange. Yeah.
So the irritant receptors cause you to, when you take gas, you stop breathing.
It's you're sucking in air against what feels like a constricted lung.
So, so the gas that you breathe in, it's like kind of in response to the bronchoconstriction.
Yeah, yeah, yeah. The pulmonary irritant receptors are located in the airways themselves, so in the epithelial lining of the airways. And then the J receptors are located also in the lining, but at the alveolar sacs. Yes?
Sorry, where are the receptors located?
In the bronchiolar smooth muscle. Yes, ma'am.
This is the blood has to develop receptors for carbon monoxide. That's why you have to breathe in.
Carbon monoxide doesn't trigger the irritant receptors. I mean, carbon monoxide is carbon and oxygen, right? So it's odorless, it's invisible. It doesn't trigger any thing. It causes a lot of problems, right? But yeah, no, it's not triggering anything. And it'd be great if it did, right? But no. Okay. Okay. So let's talk about this idea that the brain is receiving information regarding metabolic needs and using it to adjust respiration. Okay? Now, there are 3 pieces of information that is used, conveyed to the brain, and is used to adjust respiration. The PO2 of blood, so all of this is about the blood, okay? So the PO2 of blood, so the partial pressure of oxygen in the bloodstream, especially in arterial blood. Okay, so the PO2 of arterial blood, the PCO2 of arterial blood, and the pH of arterial blood. These are the 3 pieces of information. So obviously, PO2 is important because PO2 determines how much oxygen can bind to hemoglobin, which is then going to determine how much is in your blood, right? PCO2, it may not be. This is my least favorite way to lecture. It may not be as obvious why we should care about PCO2, but PCO2, the production of PCO2 directly reflects metabolic activity. Right? The more metabolically active your tissue is, the more it will produce carbon dioxide. Okay? And as we'll see when we talk about this more, your body actually primarily cares about PCO2 in order to control breathing. Okay? Your body actually does not consider PO2 very much at all under normal circumstances. Okay? Now, so PCO2 directly reflects metabolic consumption, so metabolic activity, and your blood will change in PCO2 much faster than it will change in PO2. Okay? So the CO2 levels in your blood actually fluctuate much more than your O2 levels fluctuate. Okay? So by using CO2 to control breathing, you're allowing for the adjustment of breathing before you ever see a change in O2. It's that prediction that I was describing. Okay? And then the pH is relevant because as CO2 levels rise, pH changes. Okay? But then, as we're going to talk about in this discussion, pH also has an effect on breathing for reasons that are unrelated to the control of respiration. And it's much more related to the contribution that the respiratory system has in the body's control of pH. Okay? So right now, just hold on to this idea that these three pieces of information is what the brain is using to help regulate respiration. Okay? And then the receptors in the body that can sense these things are called chemoreceptors. And we have two sets of chemoreceptors. We have peripheral chemoreceptors, and we have what we call central chemoreceptors. The peripheral chemoreceptors are located in two key places. They are found near the aortic arch and at the carotid sinus. So the chemoreceptors at the aortic arch are sensory endings that we call aortic bodies. Okay? And they are measuring the composition of the blood in the aorta. And the carotid bodies located at the carotid sinus, which is the first bifurcation of the carotid artery as it sends blood to the brain. Okay. And so the carotid bodies are assessing the composition of blood destined for the brain. Okay. So those are the peripheral chemoreceptors. The central chemoreceptors, if I jump for a moment, are actually located within the brainstem itself. So it's actually located within the respiratory center. Okay. So the same parts of the brain that are helping to control breathing, they are also contributing to the sensation of this chemoreception. Okay. So the central chemoreceptors, because they are part of the brain, do they have direct access to blood? No. Right? So the central chemoreceptors actually are indirectly measuring the composition of the blood. Okay? Because the central chemoreceptors only have access to what? CSF. Okay? Cerebral spinal fluid. Okay? So keep that in mind. Now, as we talk about this, I'm going to fill in this chart right now. And what I'm going to do with this chart is describe to you, can everybody see this chart pretty well? This is going to be a 3-star system. So 1 star is going to represent weak sensitivity to one of these 3 factors. Okay? 2 stars is going to represent moderate sensitivity. And 3 stars means go see the movie, is high sensitivity. Okay? So peripheral chemoreceptors. PO2, moderate sensitivity. Okay, so peripheral chemoreceptors are moderately sensitive to the PO2 of blood. They are only weakly sensitive to the PCO2 of blood. and they are highly sensitive to the pH of blood. All right? Now, little asterisks here for the central chemoreceptors. The central chemoreceptors, in terms of the sensitivity to all this composition of the blood, the central chemoreceptors are indirectly measuring all of these things, right? Because all they see is CSF. So they're using CSF as a way to indirectly measure what's going on in the blood. Everybody clear with that? Awesome. Okay. Central chemoreceptors, weakly sensitive to the PO2 of blood. Okay? PCO2 of blood, highly sensitive. Central chemoreceptors, highly sensitive to PCO2 of blood. And then central chemoreceptors, in terms of changes in blood pH. Anybody want to guess how sensitive they are? Not at all. Because the hydrogen ions that are in blood that contribute to the pH of blood can't cross the blood-brain barrier. Okay? All right? So the central chemoreceptors are not sensitive to blood pH at all because the hydrogen ions in the blood that are actually changing the pH can't cross the blood-brain barrier, and the brain can't see it. Okay? Great. So let's look at the control of PO2. So in terms of this graph, the control of PO2 is the peripheral chemoreceptors are moderately sensitive to PO2. Central are very weakly sensitive to PO2. So you can see the body does not prioritize the sensation of PO2 in terms of respiratory control. Okay? It does not prioritize it. It sees it as an emergency life-saving system. Okay? So it doesn't use it to adjust breathing on a moment-to-moment basis. So the peripheral chemoreceptors are really the primary way that our body does sense PO2 when it does sense PO2. But arterial PO2 has to get to about 60 millimeters of mercury or below before the peripheral chemoreceptors even respond or care. So what's normal arterial PO2? 100. And these are numbers you should know, okay? It's fine to not know them today. Next Tuesday? No. So you have to get a 40% drop in arterial PO2 in order for the peripheral chemoreceptors to really do anything about it. Okay? Now, to get some perspective on this, 60 millimeters of mercury, if you looked at that dissociation curve for hemoglobin, 60 millimeters of mercury is about 92% hemoglobin saturation. Okay? So 60 millimeters of mercury is about 92% hemoglobin saturation. So even with a PO2 drop of 40%, right, your hemoglobin's still pretty saturated. However, yeah, you're at the cliff, right? 92% represents the cliff. So the peripheral chemoreceptors are like, all right, I can't ignore this anymore. We're at the cliff, right? So at that point, the peripheral chemoreceptors are going to trigger respiratory stimulation. So it stimulates the medullary respiratory center, stimulates an increase in the rate and depth of breathing in order to attempt to elevate arterial PO2 back to normal. Okay? So this is really seen as kind of a life-saving measure, an emergency system. Okay? It does not contribute to the normal moment-to-moment regulation of respiration. Okay. The central chemoreceptors, in terms of the control of PCO2, the central chemoreceptors are really the guys to do this job. Okay? So carbon dioxide in the blood, if you recall, I'm just going to, this should say 60, not 30. Okay? So that when we look at the ways that blood carries carbon dioxide, physically dissolved in plasma is about 15%. Bound to hemoglobin is, I don't even remember, but bicarbonate, what's that? 30. That's where the 30 came from. And then bicarbonate is 60%. Okay? Now, the PCO2 of blood, the PCO2 of blood represents what? Of those three ways that the blood carries CO2, number one, right? The amount that's physically dissolved in plasma. So the PCO2 represents what's physically dissolved in plasma. But just like we talked about with oxygen, CO2 has to physically dissolve in plasma before it can get into the red blood cells and bind to hemoglobin or form bicarbonate. All right? Yeah. So we're talking about measuring the PCO2 of plasma. Okay? So the PCO2 of plasma is an important number because number one, it represents the amount of carbon dioxide physically dissolved in plasma. Okay, that's all it reflects, is the amount that's physically dissolved in plasma. But the reason why it's so important is because CO2 has to dissolve in plasma before it can then be carried in any of these other two ways. Okay? Now, the brain, the central chemoreceptors in the brain, the CO2 that's dissolved in plasma easily crosses the blood-brain barrier. Okay? So the CO2 that's dissolved in plasma easily crosses the blood-brain barrier. But the CO2 that's bound to hemoglobin, is that going to cross the blood-brain barrier? No. The CO2 that's found in the form of bicarbonate, no, because it's charged, right? It's not going to get into the blood-brain barrier. So the CO2 that's dissolved in plasma can cross the blood-brain barrier, and once it's across, those central chemoreceptors can sense it. Right? Now, I'm going to say something and you can immediately ignore it if you want to. Once that CO2 does cross the blood-brain barrier, once it's across the blood-brain barrier, it can actually then form bicarbonate in CSF, right? And it can affect the pH of CSF. Make sense? And the central chemoreceptors are actually using even slight changes in CSF pH as their direct means of measuring PCO2. Okay? So the central chemoreceptors are indirectly measuring blood PCO2 by directly measuring the pH of CSF. Okay? So I'm going to say it again, but again, you're going to ignore it if you want to ignore it. But the central chemoreceptors indirectly measure blood PCO2 by directly measuring any slight changes in pH of CSF. Okay? But what you should absolutely remember is that the central chemoreceptors are highly sensitive they are highly responsive. This is the best way to put it. They're highly responsive to even slight changes in blood PCO2. Okay? So the central chemoreceptors are highly responsive to even slight changes in blood PCO2. All right? Okay. Now, when arterial PCO2 increases, okay, the central chemoreceptors sense that change and stimulate the medullary respiratory center. And the response is, if PCO2 increases, the response is an increase in the rate and depth of breathing. And that increase in rate and depth of breathing blows off that CO2 and brings it back down to normal. Okay? And those changes in PCO2 sensed by the central chemoreceptors is how we regulate breathing on a moment-to-moment basis. Okay? Now, if, however, your PCO2 becomes extremely high, okay, if PCO2 becomes extremely high, now there's sort of two ways this happens. It can happen gradually, and the PCO2 can become very high chronically, like you see in obstructive pulmonary diseases like emphysema. Right? Their PCO2s are through the roof. Okay? Now, in that case, what happens is the central chemoreceptors actually decrease in their sensitivity. So they stop responding. Okay? So that's one effect of chronically high PCO2 that sort of gets there over time. When it happens acutely, when it's a very rapid change and PCO2 becomes very high, that actually represents injury to the brain, and you get suppression of respiratory function. And basically that interferes with the ability for the respiratory center to function, and ventilation goes down. This is not compensatory, right? This just represents the injury that acute extreme elevations in PCO2 do to the brain. Yes, ma'am?
What might cause an acute increase in PCO2?
Acute respiratory distress, right? So things like you just stop breathing altogether for a period of time. You're unconscious and you don't just stop breathing entirely. Anything, acute injury to the sites of gas exchange, right, because of smoke inhalation or massive inflammatory, injury. So these acute injuries that interfere with respiratory effort. Yeah. Yes, high CO2 levels are causing the injury.
But in both situations, the chronic and acute receptors stop responding.
Yeah. Yep.
You said the central chemoreceptors don't respond to pH, but
Of blood.
Of blood.
Of the blood.
But it's no response to pH of the CSF. So that's just about blood. But it doesn't look at the blood ever.
It doesn't directly look at the blood ever. So it can,
When I look at
yeah.
this, I think,
So the reason why it can see what's happening in the blood in terms of PCO2 is because CO2 can cross the blood-brain barrier. So a change in the PCO2 of blood is going to cause a change in the PCO2 of CSF.
right, that I understand. What I don't understand, I'm just reading the chart, what I don't understand is peripheral chemoreceptors are very responsive to pH in the blood. Chemoreceptors, central chemoreceptors can't respond to pH in the blood because they're not near the blood. They're only in the CSF. But they are responsive to pH just in the CSF.
Yes, because changes in the pH of CSF are entirely carbon dioxide derived.
And not, so you could have a completely different pH in the blood and a different pH in the CSF. Are they related to each other in some?
Only if it's CO2 derived. So we're going to get to this, but basically, and you guys may have already talked about this in some of your classes, but when you talk about a change in the pH of the body, you talk about it in terms of an acidosis versus an alkalosis, and there's a difference between a respiratory acidosis, for example, versus a metabolic acidosis. And a respiratory acidosis is caused by an elevation in CO2 and an elevation in CO2-derived acid in the blood. But a metabolic acidosis is caused by non-respiratory acid. Right? So if you have an acidosis in the body that is unrelated to CO2, your central chemoreceptors can't see it, but your peripherals will. And we'll get into that, so I'm not going to answer questions right now about that, but yes, sir. Yeah. Yeah, so and we talk about this in patho, but basically for those patients that habituate to hypercapnia, they habituate to elevated carbon dioxide levels, carbon dioxide no longer controls their breathing. So when carbon dioxide no longer controls your breathing, what's left? Oxygen, right? And for a patient who's relying on oxygen and the peripheral chemoreceptors to control their breathing, right? That means if their hemoglobin saturation, if their PO2 gets above 60 and their hemoglobin saturation gets above 92, what happens to them? They stop breathing because there's no longer any drive for them to breathe. So for these patients that have emphysema and have become habituated to hypercapnia, actually over-oxygenating them causes problems. They stop breathing. So they actually have to live right at the edge because if their PO2 gets above 60, their drive for breathing stops. So we'll talk more about that in patho. Yes, ma'am. Last question.
Earlier, you were talking about goals for breathing. You said that the constant interviews from my lungs, that's basically making normal respiratory rate. But then with the apnesis that you mentioned, with the gas for that consideration, you said that there was damage to the apnesis receptor or the pneumotaxis receptor, which is part of the cause.
There's two things happening with apnoesis. There's damage to the pneumotaxic center, so it's not contributing to it, and there's some sort of interference with the ability for the brain to receive that feedback on metabolic activity or interpret it. There's both things are happening. Okay? All right, so finally, there's this issue that changes in blood pH will affect respiration, okay? Because the peripheral chemoreceptors are highly sensitive to blood pH. Okay? Now, when we look at arterial pH, which is a measure of hydrogen ion concentration, okay? hydrogen ion concentration in circulation comes from two sources. Okay, so arterial pH comes from CO2-derived acid in the form of carbonic acid. Okay, so as CO2 levels rise, acidity is going to rise. But the other place that acidity comes from is non-CO2 derived, okay, which is completely sort of lumped into what we call metabolically derived acid. Okay. So the overall arterial pH is going to be determined by the level of CO2 derived acid and non-CO2 derived acid. Okay. The peripheral chemoreceptors are sensitive to pH, and they don't distinguish between CO2 derived and non-CO2 derived. Okay. Any drop in pH, any acidosis of the bloodstream stimulates the peripheral chemoreceptors, and it stimulates breathing. Okay. Now, when that happens, let's say the problem is CO2. Let's say your respiratory rate goes down for some reason, you're not breathing enough, okay? Your CO2 levels rise, okay? Before a rise in CO2 can actually affect the blood pH, that rise in CO2 is very quickly going to be sensed by those central chemoreceptors. Okay? So if there is a rise in CO2, the central chemoreceptors are very likely going to respond before you get a really significant change in overall blood pH. Does that make sense? So a rise in CO2 is predominantly going to be addressed by the central chemoreceptors. Now, if your pH becomes acidotic, if your blood becomes acidotic, let's say you have type 1 diabetes. It's uncontrolled. Your body can't get glucose into your cells, so it starts using stored fat as an energy source. And you just are breaking down stored fat like crazy. And the byproduct of fatty acid metabolism is the production of ketones or keto acids. So you go into something called ketoacidosis. Now that's an acid that's metabolically derived. Okay? Patients like that, and you've probably read this question already in the study guide, they tend to exhibit this thing called cusmal breathing, which is basically a hyperventilation. Okay? What are they doing when they're hyperventilating? They are blowing off CO2. And that breathing is a compensation. It's an attempt for them to reduce their CO2-derived acid because they have an elevation in metabolic acid. Okay? So the hyperventilation that happens during metabolic acidosis, like with these diabetic patients, that hyperventilation is blowing off CO2, trying to decrease CO2-derived acid in order to compensate for an elevation in metabolic acid. Okay? And in medical circles, that's referred to as a respiratory compensation for a metabolic acidosis. Okay? So in this way, and that's triggered by the peripheral chemoreceptors. So in this way, the peripheral chemoreceptors is adjusting breathing not for the sake of respiratory control, but rather for the sake of pH control. Okay? That makes sense? Okay, a couple of questions. Yep.
So the peripheral chemoreceptors are adjusting, I guess what I'm trying to get at is between compensation if it's an increase in arterial CO2 or not. If it's an increase in ventilation or not, but then it's adjusted, how's it adjusted for arterial CO2 again?
So if non-CO2 derived acid is normal, and you're just getting a rise in CO2, a rise in CO2 is going to trigger the central chemoreceptors before it causes enough of a pH change to trigger the peripheral. A rise in PCO2 in the blood is going to trigger the central chemoreceptors before it changes arterial pH enough to trigger the peripheral. Okay?
So a rise in arterial pH, that happens, the central chemoreceptors will adjust before the peripheral chemoreceptors can sense that.
I'm saying that a rise in CO2 in the blood is... So a rise in CO2 in the blood is not going to cause an immediate rise in acidity of the blood, okay? Because we have all these buffer systems in the bloodstream, a rise in PCO2 has to be above a certain threshold for it to cause a change in pH and trigger the peripheral chemoreceptors. And within that range, the central chemoreceptors have already taken care of it. Does that make sense? Yeah.
If the central chemoreceptors are responding preemptively, are you still going to have the hyperventilation even if it's not exactly the Kussmaul breathing? Is there compensation still going to be increased ventilation?
The central chemoreceptors are only going to respond if there's a rise in CO2. In the diabetic, the central CO2 starts off normal, and then the Kussmaul breathing brings it below normal.
So there's never actually a change.
So in Kussmaul breathing, the person's respiration is normal. So their CO2 is normal. But because their blood pH is acidic, that triggers the hyperventilation. So their CO2 goes from normal to low to compensate for the acidosis. Yes, ma'am. Don't worry about the circuitry. So the central chemoreceptors stimulate breathing. That's all you need to keep in mind. It involves all of it. Don't worry about circuitry. Okay, so one more question. Yep.
So the peripheral chemoreceptors are going to be more responsive to metabolic acidosis, where the central chemoreceptors will be responsive for respiratory acidosis.
So the central chemoreceptors are going to be completely unresponsive to metabolic acidosis. Okay, that's number one. What's detecting hydrogen and not CO2? Yes, yes. So the central chemoreceptors are going to be completely unresponsive to metabolic acidosis. And for a respiratory acidosis, both receptors are going to be lighting up like crazy. But think about this. If you have a problem with the respiratory system, enough for you to have respiratory acidosis, even though these receptors are lighting up like crazy, the respiratory system can't correct it, right? So and so this is the next story which we talk about in patho, is that at that point for a respiratory acidosis, we get a metabolic compensation. Okay? Yeah. Okay. So, Sarah, the question, right? In the study guide question, the last study guide question, it's something to the effect of she holds her breath and finds out she can hold her breath for, let's say, a minute and a half, okay? And then she discovers that if she hyperventilates, okay, and then immediately holds her breath, she can hold her breath for 3 minutes. She can hold her breath for twice as long, okay? So the question is, why can she hold her breath longer the second time? What did she do by hyperventilating? She blew off CO2. So she lowered her PCO2, so it took a longer time for her PCO2 to get high enough for her central chemoreceptors to trigger breathing. Okay? And during all of that change, her PO2 never budged, right? The PO2 of her blood never changed, and the hemoglobin saturation of her blood never changed. Okay? So that breath-holding experiment, and this also gets into the difference between voluntary control of respiration and involuntary control. So you certainly can voluntarily hold your breath. Your brain doesn't think that's a great idea, right? But hey, you're your own person, you can do it. But at some point, your brain's going to say, all right, you are clearly not responsible enough to take care of this, so we're going to take over, okay? And so you can't hold your breath to death, right? Because at some point, your PCO2 is going to get high enough, it's going to force you to breathe. The year I moved back to New York, that was like, it's like a year after. I think it was like 2004, 2005. Does anybody know David Blaine? Yeah. So around like 2005, he did this like just ridiculously stupid thing in front of Lincoln Center. Does anybody know what I'm talking about? So he spent a week in this like bubble of water. And so his whole, he does these, David Blaine is a illusionist, he's a magician, and he, just in recent years, he's really gotten into sort of extreme physical deprivations. So his plan was to spend a week in this exposed bubble of water, and then at the tail end of that, he was going to break the record for holding his breath underwater, okay? And because that's just not exciting enough, they shackled him, right, like Houdini. And he was going to get out of the shackles and then, hold his breath and then swim up. And I think the world record was like, I want to say it was like 15 minutes or something like that. Yeah. Yeah. So, or maybe it was 8 minutes, I don't remember. But it was a long time. So, what happened, he didn't make it, right? So, so what happened was, he really underestimated what being in water for a week was going to do to the body, especially in a globe that like magnified the rays of the sun into his little world. And then he also underestimated how much energy he was going to expend trying to get out of the shackles, right? So what happened was he's sitting in the bubble, and you can see him sort of starting to struggle. And a couple of air bubbles escape from his mouth, right? And then immediately, like a team of scuba people dive into the bubble, right, and rescue him. And what they do immediately when they catch him is they grab his mouth and nose, right? Because what was he about to do? He was about to take in a huge breath, basically a huge gulp of water right into his lungs, and he was about to drown. So that's what happens when people drown, is that their respiratory center, they're not shackled, and but basically the respiratory center just forces them to take in and attempt to breathe, and water fills their lungs. Okay? And so you see his team sort of grabbing his face and pushing him up to the surface so that he can actually take in that breath of water. And he did an interesting TED Talk, which I'll post to the discussion board, talking about that and then talking about his foray into extreme breath holding. And how he kind of rebounded to actually eventually break the world record, eventually. But and you can tell in the video he's done some damage because he's really not right in the head. But it's really fascinating. But he talks about this is called like a static breath hold, meaning that you are trying to be as still as possible and holding your breath for as long as possible. And that basically the technique requires people to expend as little energy as possible, so they're producing as little CO2 as possible and consuming as little oxygen as possible. And then they also do this crazy meditative training to drop their heart rates as low as they can. So they're actually, and they do this purging, meaning that they hyperventilate and get rid of as much CO2 as they can. So there's all these crazy techniques to trick your brain into not triggering you to breathe, right? But it's a fascinating kind of look at what he goes through and learns all these different techniques. Yeah. Yes. It's PCO2. It's PCO2. Yeah.
So I thought that you had said that the PCO2 and something else wasn't
PO2. Yeah. So your PCO2 of arterial blood is like 40, right? And so you're starting off at 40, and then when you're holding your breath, you're not getting rid of that CO2. So the CO2 production continues, so your PCO2 starts to rise, and then eventually it pushes you to breathe. But if you hyperventilate, you can get it from 40 to let's say 38 or something. So it takes longer to hit that threshold. That's really what that's about. You're welcome. Okay. Yes, ma'am. One more question, and we're going to move on.
So this response to metabolically derived acidosis, is it like a short-term response, or is any high acidic level going to cause the
Is it a short-term response? It's not short-lived, right? I mean, you're going to continue to your respiratory rate is going to continue. And it's not going to necessarily fully compensate for the acidosis. So you're eventually, if you don't have some kind of intervention to reduce your acidosis, the acidosis is really the bigger problem. Does that make sense? So the fact that you're hyperventilating constantly is not really an issue. It's really that you have this acidosis that needs to be corrected. Does that make sense? You have low body pH, then you have a pathological problem. It's a disease. It's not your body maintains pH between 7.45 and 7.35, and if it gets below 7.35, you're acidotic, and that's never a normal thing. So, yeah, if it's a metabolic acidosis, yeah. So, depending on how severe the acidosis is, the respiratory rate, instead of it being 12, it might be 16 or 18, right? So they might not look like they're just breathing super hard, right? Unless the acidosis gets pretty severe. But it's going to constantly stimulate breathing until that
that pH is corrected.